Hello, this is Dr. Ahmed Majid Hamza Al-Shimari, the head of Experimental Therapy Department at the Iraqi Center for Cancer and Medical Genetic Research. Today, I am presenting our work about detection of a neural stemness status through a neurogenesis process. This work was done with my colleagues Dr. Maida Mohammed, Dr. Naif Yasin, and Dr. Ahmed Adnan Al-Juburi. The paper is entitled Characterization of a neural stemness status through a neurogenesis process for bone marrow mesenchymal stem cells. The in vitro isolation, identification, differentiation, and neurogenesis characterization of the mesenchymal stem cells were investigated to produce two types of cells, neural cells and neural stem cells. These types of stem cells were used as a successful source for the further treatment of central nervous system defects and injuries. The mouse bone marrow mesenchymal stem cells were used as a source for stem cells in this study. Β-Mercaptoethanol was used as the main inducer for the neurogenesis pathway to induce in neural cells and to identify neural stem cells.
Methodology: isolation of mesenchymal stem cells from mouse bone marrow, culturing and propagation of these cells, immunophenotypic analysis of mesenchymal stem cell markers, followed by starting neural differentiation induction by β-Mercaptoethanol, and evaluation of the neurogenesis process by studying the neural marker by immunocytochemistry study and by real-time PCR quantification assay. Three types of neural markers were used: nestin as the immaturation stage marker, neurofilament light chain as the early neural marker, and microtubule-associated protein 2 as the maturation marker through different time intervals in the neurogenesis process, starting from the mesenchymal stem cells as undifferentiated cells toward neuron cells as differentiated cells. The results of different exposure times to β-Mercaptoethanol of the neural markers analysis done by immunocytochemistry as shown in this figure and by real-time PCR assay helped us to identify the exact timing of the neural marker stemness state. This figure showing mesenchymal stem cells after induction by β-Mercaptoethanol as shown as under inverted microscope for different time intervals. The result showed that best exposure time that may be used for the production of neural stem cells was 6 hours, and this is confirmed by real-time PCR assay as shown in this figure by measuring the neural marker through different time intervals. This figure showing neural stem cells induction by 6 hours, figure A, and figure B after 24-hour exposure to β-Mercaptoethanol as revealed under inverted microscope. The best maintenance media for neural stem cells were also identified. Furthermore, we optimized exposure to β-Mercaptoethanol with different times and concentration, which could be an interesting way to modulate specific neural differentiation and obtain autologous neural phenotypes. As shown in this figure, we used 4 different formulae to propagate neural stem cells. The 4 different formulas were evaluated by real-time PCR assay to confirm which formula was the best for maintaining neural stemness status, where nestin was constantly expressed through 2 passages, as shown in the figure. This figure showing the neurogenesis pathway that mesenchymal stem cells induced toward the neurons, where we can indicate the neural stemness state by following gene expression of the neurogenesis marker, nestin, neurofilament light chain, and MAP2 after proof of stemness at 6 hours of exposure to β-Mercaptoethanol. This study was able to characterize the neural stem cells in culture under differentiation for neurogenesis in the pathway of the neural differentiation process by studying the expressed neural genes and the ability to maintain these neural stem cells in culture for further differentiation into thousands of functional neurons for the treatment of brain and spinal cord injuries and defects.
Thank you for listening. Thank you for interest in our paper. Thank you very much, and bye-bye.